plenty of time to finish it then. So I will only quiz you over the part that I covered today. So anything that I didn't get to will not be on the quiz for next week. Okay? Let's begin with prayer. Father in heaven, thank you that we could be together this morning. I'm so thankful for each of these students and their hard work and their dedication. And help us to have a nice, restful weekend that's coming up. And bless us during this time of study now, we pray in Jesus' name. All right, so we were talking about liver. We had just talked about the 3 different ways of looking at liver lobules. We talked about the classic lobule and the portal lobule and the liver acinus. So make sure you understand those concepts. There will probably be a quiz question next week covering that, although I'm not sure, but very likely. All right, we're going to look now at some of the other structures of the liver. We're going to look at the vasculature of the liver, and we're going to look at the liver cells and what they do. So first let's talk about sinusoids. Sinusoids are large diameter capillaries, and so they're lined by endothelium, as all vessels are. But the liver sinusoids are different from a typical blood vessel, so let's talk a little bit about them, if I can get my remote to work here. I guess my remote is on strike. Let me try restarting this and see if that works. The reason why it's not working is because my little USB gilly is gone. So where did it go? All right, I will have to use the mouse. Hate using these. All right, so sinusoids are large diameter fenestrated vessels. What does fenestrated mean? Fenestra is Latin for window, which means they have little perforations in the walls. The wall is not solid, it's fenestrated. So it's fenestrated endothelium and surrounded by plates or columns or cords of hepatocytes. Remember, hepatocyte is the main cell type. Now there's another type of cell in addition to hepatocytes which we call Kupffer cells. Kupffer cells are a type of macrophage, so they're part of the mononuclear phagocytic system, along with Langerhans cells, macrophages, monocytes, osteoclasts, microglial cells. Those are all part of the MPS system. Now, there's an interesting little buffer zone which is called the space of Disa. You'll hear most anatomy or histology professors call it the space of Dissay. That's what my teacher called it. If it was French and had an accent on the E, it would be Dissay, but it's not. It's German. It's not French. And so I'm learning German on Duolingo right now, and I am an expert in German pronunciation, and so it's Disa, not Dissay. I just had to get that off my chest. All right. Now, the space of Disa is part of the hepatocyte, but it's at the interface between the hepatocyte and the sinusoid. So rather than the surface of the hepatocyte directly contacting the endothelial cell, there is a little space in between them, and this is this space of Disa. I don't know why that space needs to be there. We'll look at some pictures and hopefully it'll help to clarify things a little bit. And then there's another type of cell called the hepatic stellate cell, also called an Ito cell. And I don't know much about its function. You probably will learn more about that in your, I don't know, did Dr. Teal already talk about liver function and stuff with you? Has he gone over that?
No.
Some people say yes and some people say no. So I know which ones were in class and which ones weren't. Anyway, the Ito cell becomes prominent when there's liver disease and stuff. I'm not sure what it does, but it contains vitamin A and it has fat droplets in it, for what that's worth. You will not have to identify one in lecture, in lab. And I just hit the button accidentally, because I don't like mouses, whatever they're called. All right, so here you can see columns of hepatocytes, and notice that this is a branch of the portal vein. This is our little portal triad here, so this is a portal area. Remember, the classic lobule has the 6 portal areas, so this is one of them. And notice that the blood from the portal vein flows into one of the sinusoids, and also blood from the little hepatic artery is going to flow into the sinusoids as well. So these sinusoids carry blood from veins and arteries to the central vein. And so notice that every hepatocyte is in contact with the sinusoid. And so they all have this very intimate contact. And then the hepatocytes are also producing bile, which is going to flow in these little bile ductules to these bile ducts and then out of the liver later on. Okay, so we'll get to the bile later. So here is two electron micrographs. The one on your left is at lower magnification. And so this is the hepatocyte. Notice all the mitochondria in it, lots of them. And this is that little space of DISA. And then here is the lumen of the sinusoid. So this is a little buffer zone between the hepatocyte and the sinusoid. And here you can see it down here as well. So that's the space. Here it is at higher magnification. So notice that the hepatocyte has lots of microvilli type structures extending into this space. And then here's the endothelial cell, which borders on the space of DISA. So fluid from the plasma in the sinusoid can leak out into the space of DISA, and then secretions from the hepatocyte are released into the space of DISA. So it's sort of a buffer zone between the two areas. I don't know why that space has to be there. The only thing I can think of is that all these little microvilli type structures increase the surface area for the release of more substances that the hepatocytes are secreting. I probably should look into that and have a better answer than I do. All right, now in addition to the blood flowing toward the central vein, we have bile being produced by the hepatocytes, and bile is going to flow in the opposite direction as the blood. Because the blood is coming into the liver, bile is going out of the liver, and so we, at each one of those little corners, the little portal triads, we have a little tiny bile duct. And so each hepatocyte has a little bile canaliculus between it and its neighbor, a little tiny canal into which it releases its bile. And then the bile flows into small ducts and bigger ducts and bigger ducts and so forth. So it starts out in these bile canaliculi, which are tiny little spaces between adjacent hepatocytes. I'll show you a picture in a minute. And then those flow into larger vessels called cholangioles. Angi, that part of the word, refers to vessel. Remember, angiogenesis is vascular development, right? And angioplasty is when you stick something into a blood vessel and plaster the plaque against the wall so you open up the blood vessel, angioplasty. So anything with angi in it refers to a vessel. Chol refers to bile. Remember cholecystokinin, gallbladder, cholecyst. So cholangiole would mean a bile vessel or a bile duct. So a cholangiole is a small duct. And the cholangioles will empty into larger vessels, which we call canals of Herring, whoever Herring was, I have no idea. And then those drain to larger vessels or larger ducts, which we call interlobular ducts. So obviously between the various lobules. And then those are going to merge into larger ducts that lead to the right and left hepatic ducts, which then combine to form the common hepatic duct, which then joins the cystic duct to form the common bile duct, which then joins the pancreatic duct at the ampulla of Vater, or the hepatopancreatic ampulla, and then dumps into the second part of the duodenum. Get all that? I should be recording this, shouldn't I? So let's look at this picture now. So these are the individual hepatocytes. Between the hepatocytes, we have these little canaliculi. They drain to larger vessels, which we call cholangioles, which drain into the larger vessel called the canal of Herring, which leads to a larger bile duct, which leads to the left and right hepatic ducts, which drain out of the liver. Okay, so there you go. All right, this is a picture, this is a drawing of a hepatocyte, and notice that there are several different domains around the hepatocyte. So parts of the hepatocyte border on other hepatocytes, and parts of the hepatocyte are going to border on sinusoids by way of the space of DISA. Okay? So here, notice that you have a hepatocyte here. Here's another hepatocyte here. And so between the two hepatocytes, you have this little bile canaliculus. So that's where the hepatocytes are producing the bile. They release the bile into those little canaliculi, and then those drain to the cholangioles and the canals of Herring and so forth. All right? So the hepatocytes are polygonal in shape. They're about 20 to 30 micrometers in diameter. You don't have to memorize that number. The membrane has a lateral domain, which is where you find the bile canaliculi. So the lateral domains are regions where one hepatocyte will contact another hepatocyte. Okay, so it's a hepatocyte-hepatocyte junction. That's what the lateral domain is. And there's always a bile canaliculus between the two cells. And so these are isolated by what we call fascia occludantes, or it's similar to a zonula occludens. It's just not as all-encompassing as a zonula occludens. And there are also gap junctions between the hepatocytes. There are microvilli, which increase the surface area. So you can see in the canaliculus, there's these little, what look like little dots. Those are the little microvilli. The walls of the canaliculi are rich in sodium, potassium, ATPase. They have adenylate cyclase, so pumping type mechanisms are going on. Then we have the sinusoidal domains. So this is where the space of DISA is located. And notice that there are microvilli, as we saw in the electron micrographs, projecting into the spaces of DISA. You can see that here. And then this is where various secretions, not bile, but hormonal secretions by the hepatocyte are going to be released. Because remember, hormones are released into the bloodstream, not into ducts. So ducts would be exocrine. Bile is an exocrine secretion. And then we have some hormones produced by the liver that are endocrine secretions. So they're going to be released into the bloodstream, but they release into the space of DISA, and from there they go into the sinusoid. Okay, so there's an exchange in this space between the blood and hepatocyte within that space of DISA. All right, so what organelles are prominent in hepatocytes? The nucleus is typically large and spherical and euchromatic, as you see here. Sometimes you can have two, and sometimes hepatocytes can be polyploid. Rather than being diploid, they can be triploid or up to 64N. Okay, lots of free ribosomes, lots of rough ER, lots of Golgi, so they're producing lots of proteins, some that are staying in the cell, some that are exported from the cell. Up to 2,000 mitochondria per cell. So the hepatocytes that are nearest to the central vein in a classic lobule have the most mitochondria for whatever reason. So they must have more metabolic demands than the others do. Lots of endosomes, lots of lysosomes, lots of peroxisomes. Why all these things? Because hepatocytes are one of the main cells in the body that regulate metabolism of carbohydrates, peptides, fats, and so forth. And so there's a lot going on in the hepatocytes. They're taking in nutrients from the blood that are products of digestion, and then they are processing them. This is where we have gluconeogenesis, glucogenolysis, all those kinds of processes are going on in the hepatocytes. In addition to that, there's a lot of smooth endoplasmic reticulum because this is where detoxification of drugs and various toxins occurs. So as we said the other day, if you take in too much of certain types of medications or toxins, they can destroy the liver because they overwhelm it, can't keep up, it destroys the hepatocytes, and then your liver no longer functions, and you no longer function without a liver. All right, in addition to the various organelles, we also have some inclusions. There's lots of lipid droplets, mostly VLDLs. I'm assuming you know what that is. Very low density lipoproteins. There's glycogen, beta particles near the smooth ER, and so forth. All right, so what does the liver do? It manufactures bile between 600 and 1,200 milliliters per day. The bile contains water, bile salts, phospholipids, cholesterol, bile pigments, and immunoglobulin A. And bile salts. Now, bile salts are reabsorbed in the small intestine. So bile is released into the small intestine, in the duodenum, and it participates in digestion of fats, because what it does is emulsifies the fats and breaks fats down into smaller droplets so that there's more surface area for the lipases to attack the fats. But then a lot of the bile salts get reabsorbed and sent back to the liver again. So there's a cycle that occurs. Now, some bile salts are formed de novo, that means from scratch, not recycled, in the smooth endoplasmic reticulum of hepatocytes. So you have two sources of bile salts, one by the reabsorption of salts in the small intestine, and then sent back to the liver and recycled, but you can also make new bile salts in smooth endoplasmic reticulum. We also have bile pigments. One of the main bile pigments is bilirubin. Now, some of you may have children. A lot of you probably don't have children yet. You will someday. You might recall that when babies are first born, lots of times the liver isn't fully functional yet, and so lots of newborn babies are jaundiced. And they have high levels of bilirubin in their blood, and if this is not taken care of, it can actually cause nerve damage or brain damage. And so what they will do to these babies is expose them to brief bits of ultraviolet radiation, because that breaks down the bilirubin and helps. Now, I lived in Africa for 6 years, and I had 2 children that were born in Africa, so I tell everybody I have African-American children, which is true. They just don't look like they're African-American. But anyway, I have 1 daughter and 2 sons. My daughter, a month after she was born, we went camping by this lake. And in Africa, they don't have the latest in medical technology, so she was a little jaundiced, and we didn't have bilirubin lights to put her under. So we decided to give her a little bit of sunlight, because it has ultraviolet radiation, to break down the bilirubin. Unfortunately, we left her a little too long in the sun, and our poor little baby got sunburned at 1 month old. And at the same time, this is horrible, I shouldn't be telling myself. So we were cooking some macaroni over this open little fire, and my 2-year-old son was anxious to see if the macaroni was done cooking yet, so he went to open it, and he pulled it on himself and scalded his foot with his boiling water. So I grabbed him and stuck him in the lake, not under the water, but his feet. And he had these horrible burns in his feet, so I had to take him to the hospital. It wasn't a fun camping trip, let's just put it that way. He survived. I don't know if he still has scars or not. I haven't looked at his feet lately, but he's going to be 40 years old next year. He's freaking out. This is a long time ago. What does that have to do with bilirubin? Not a whole lot. But there's also biliverdin, phycobilin, so there's a whole slew of these bile pigments, and many of the pigments are breakdown products of hemoglobin. So remember that the spleen, I guess we haven't talked about that yet, the spleen breaks down old red blood cells, and the hemoglobin in the red blood cells is split up, and part of the byproduct of this split up of hemoglobin is bilirubin. So that's where some of these pigments come from. This picture is just showing the recycling of bile salts, so secretion of bile salts, of bile acids and bilirubin. I guess not the reuptake, but you can read this for yourself. And what else does the liver do? It's involved with metabolism of various organic substances, so lipids from the digestive system enter the lymphatic system, and then they reach the liver by the hepatic portal artery, or the hepatic artery. They're broken into glycerol and fatty acids. The fatty acids are used to make phospholipids and cholesterol and so forth. And the acetyl-CoA is used to make ketone bodies. And hepatocytes manufacture VLDLs, and hopefully you've had all this stuff in biochemistry or physiology, because that's all I need to say about it. All right, carbohydrates. The liver helps to maintain normal blood glucose by taking in and storing glucose as glycogen, if there's excess glucose, by reforming glucose by breaking down glycogenolysis as needed, and making new glucose from other sources by way of gluconeogenesis. So all of these processes take place in the liver in order to regulate glucose levels in the blood. Proteins, hepatocytes manufacture about 90% of blood proteins, and this includes clotting factors, complement proteins, transport proteins, albumin, and they also manufacture globulins, all of them except gamma globulin, and manufacture non-essential amino acids. You know the difference between essential and non-essential? What's the difference? Our body doesn't make essential amino acids. That's why they're essential. We have to obtain them through eating. But other amino acids our bodies make, so they're not essential. And then converts ammonia to urea. So here's just another picture showing glucose metabolism and so forth. You can look over that in your leisure. All right, other roles: storing vitamins, degrading hormones by lysosomal enzymes, degrading drugs and toxins. And there's several different methods of degrading them: methylation, conjugation, oxidation, blah, 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 blah. And then immune function. So the liver produces IgA and releases it into the bile, and then the Kupffer cells are phagocytic cells, so they're part of the immune system as well. All right, so that's it for pancreas and liver. I went through that fairly rapidly. I will sort of, it's always good to read a little outside of the PowerPoint to get a deeper understanding, but I'll base my questions on information from the PowerPoint. I also post handouts for most of my lectures if you've discovered those. They contain much of the same information that's in the PowerPoint, but some people like a Word document to study from rather than the PowerPoint, so they're there as well. All right, now we're going to switch and look at salivary glands.